Hello, my name is Nain de la Cruz, and in this presentation, I'm going to share with you the preliminary results of our work titled Analysis of Shannon Entropy and Mutual Information in the p53 signaling pathway in response to radiotherapy. This work was done in collaboration with William Albornoz and Jesús Rodríguez González from the Centro de Investigación y de Estudios Avanzados del IPN, Unidad Monterrey. The content of the presentation is divided as follows. First, we present the introduction. After that, we show the methodology we use, and then discuss the results, and finally, we provide the conclusion and further work for our research. We begin the presentation saying that inside the cell, there are complex processes that lead to different outcomes, such as cell cycle arrest, DNA repair, senescence, and apoptosis. The molecular interactions inside the cell is believed that are affected by biological noise and crosstalk. However, then we have the following question. How the outcomes remain so robust despite this biological noise and crosstalk? Biological noise refers to the variability that exists in the expression of certain protein, and crosstalk refers to the different interactions that the same molecule might have with other molecules. So we would like to understand how this robustness is achieved. However, understanding this reliability requires tools that are beyond traditional statistical tools. Therefore, we propose that information theory might help us to quantify and evaluate how these signals are transmitted effectively between molecules. When we analyze the literature, we find the following limitations in traditional statistical tools. For example, correlation quantifies covariation, but cannot help us to identify causality or discriminate between variables. In the case of regression models, this technique fails when there is non-linearity in the interactions of the variables, when there is feedback or when there is a switch-like dynamic. Besides, these methods rely on a priori assumptions, such as linearity, which limits the ability to detect several dependencies. Some alternative methods include principal component analysis, which captures the variance. However, it is not possible to detect complex signaling interaction. In the case of clustering, it becomes unstable in noisy or high-dimensional data. Network topology is a tool that gives descriptive results but not quantitative results, and information theory allows to quantify dependencies without assuming linearity. Therefore, we choose information theory as the tool to analyze the data we obtain from the molecular interactions. Now, at the beginning, we thought of analyzing the p53 signaling pathway using these techniques of information theory. The reason for this is that the p53 signaling pathway is in charge of initiating the DNA repair, and in case the DNA cannot be repaired, it initiates the apoptosis. However, the interactions between molecules inside the signaling pathway still remain unclear, and we believe that it is necessary to see how the noise from protein variability and crosstalk affects the signal transmission. Also, we would like to see how the effects of a certain level of radiation might affect the information flow between the components of the signaling pathway. For example, we would like to see if the behavior of ATM is correlated to the behavior of WIT1 or vice versa, just to mention one example. Therefore, in our work, we propose to analyze the response of cells to radiotherapy using Shannon entropy and mutual information. And with this, we would like to quantify the uncertainty in the response of three elements of the pathway, which are double-strand breaks of DNA, ATM, and WIT1. And we would like to determine if they are correlated somehow. To do that, we are going to apply Shannon entropy to measure the uncertainty and mutual information to infer relationships and information flow. In order to generate the data necessary for the analysis, it's necessary to have a mathematical model. We chose the mathematical model from reference 14, which we present in the figure. This mathematical model is divided in three different blocks. The first block is the nuclear block, which includes proteins such as p53, MDM2, ATM, as others. The other block is cell cycle arrest model, which is the model that initiates the cell cycle arrays and DNA repair. And finally, the apoptotic model, which includes caspases and is the model that allows the triggering of the apoptosis of the cell. So initially, this model include 33 differential equations, but in order to be useful for us, it was necessary to convert it to an stochastic model, and to do that we use the Gillespie algorithm. To conduct the analysis, it was necessary to induce different levels of radiation, and also it was necessary to generate different trajectories. For the present work, we generated 10,000 simulations for each of the proteins. After we got the data, now we introduce the mathematical tools we use. Graphically, these mathematical tools are represented in the figure. The whole blue circle represents the Shannon entropy, uh, and the orange circle represents the mutual information. So if the variables are related somehow, there's going to be an intersection between the two circles, which represent the input and the output. But if there is no some relation between them, then there's not going to be intersection. So in order to quantify the Shannon entropy, we record to equation number one, and if to find the mutual information, we use equation number two. So the results of the Gillespie algorithms are shown in the following figures. We only plot or we only present in this presentation three different results, one for 1 gray, another for 4 gray, and the last one for 7 gray. And we only focus on three different elements which are useful for preliminary analysis, which are double-strand breaks of DNA, ATM, and WIT1. In the horizontal axis, we see the time measured in hours, and in the vertical axis, we see the molecules amount of each of the proteins that we are analyzing. From this first figure, we can see that there is a high variability in the proteins. So when we increase the level of ionizing radiation, we see a decrease in the expression of the protein, and if we increase the ionizing radiation level to 7 gray, then we see a further decrease. But the question remains, is somehow this decreasing on the level expression of the protein somehow related between the different variables that we are presenting, or is this random at all? So in order to get an answer to that question, it is necessary to apply the um, information theory tools, which are Shannon entropy and mutual information. First, we show the results of Shannon entropy. It is important to remember that Shannon entropy only measures the variability of the proteins. For example, we can see from the figures before that the variability of the proteins was kind of high at 4 gray, and then it was reduced at 7 gray, and we might expect that the magnitude of the Shannon entropy will decrease, but then from the figure we see that this is not the case. Actually, we have the same level of Shannon entropy from 1 gray to 7 gray in the proteins of ATM and WIB1, and from 4 gray to 7 gray in the double strand break of DNA. So, uh, at the first sight, we can see the benefit of using Shannon entropy. It can rarely give us a glimpse of how the data is behaving, which means that we are having the same behavior, we have the same variability, and even if we don't see the same variability, the statistical tool is helping us to elucidate this conclusion. But is this variability related somehow between the proteins? Then we apply the mutual information. In order to apply mutual information, it is necessary to form pairs of variables. In this case, we use the following pairs: double strand breaks of DNA and ATM, double strand breaks of DNA and WIB1, and ATM and WIB1. From the results, which are also conducted at 1 gray, 4 gray, 5 gray, 6 gray, and 7 gray, we can see that the better correlation is between ATM and WIB1, and is kind of counterintuitive when we analyze the results between double strand breaks of DNA and ATM, because it is well known that ATM activates when it senses the double strand breaks of DNA, and we will expect that if we see more double strand breaks of DNA, then there is more expression of ATM. But then the analytical tool is telling us that this is not the case. Actually, there is a better correlation at low levels of ionizing radiation than at higher levels of ionizing radiation. But in the case of ATM and WIT1, we can see that there is a better correlation at 4 gray and beyond. And this hint us that there is it is necessary to carry on further research in these interactions, and this might also point out to the elements that we need to focus in order to bring different outcomes in the signaling pathways. So, with these results, we show that the tools from mutual information the tools from information theory are very helpful in elucidating the interactions of the different elements of the signaling pathway, and all the further research is necessary to be carried out. We have some idea in what direction we could go from here. So in our work, we study the variability and the interactions between three elements of the p53 signaling pathway, which were double strand breaks of DNA, ATM, and WIT1. And we conducted the study using Shannon entropy and mutual information from information theory. We saw that the Shannon entropy increases as the level of radiation increases, and although we saw a high decrease in the response of the variables in the stochastic um, in the stochastic plots, we see that this does not correspond with different levels of Shannon entropy, and the same level of Shannon entropy is kept after we increase the level of ionization radiation from 4 gray to 7 gray. Also, from the mutual information, we see that there is a better correlation between the behavior of ATM and weak one, and this points to further investigation on the type of relation and if the information flows in what direction or another. So from this, we still have the question of how perturbing these interactions might affect the behavior of the whole p53 signaling pathway, and this is something that we will carry on in future research for our work. So we would like to thank you for your presence in our presentation, and we're open to any question or inquiry you have about the content presented in the presentation. Thank you very much.